Hi everybody, welcome to the AATRN channel for another exciting tutorial. Today I will talk about DyNeuSR, which is a module for topological analysis of functional magnetic resonance imaging. So whether you are a neuroscientist excited to try new methods or a topological data analyst seeking to extend their range of applications, this is the right tutorial for you. So really thank the creators of the module, which is Genies, Borns, Petri, and Sagar, and my university, which is making this possible. Okay. So let's start with fMRI. This is a simple visualization of data. You can imagine that at each time the brain is exhibiting some neural activity. So red is a sort of positive signal and blue is negative signal. And as time goes on, this signal draws some brain patterns, some activation maps on the space of three-dimensional pixels that we are looking at. So what I hope you will be able to do by the end of the video is topological summaries like this. So you can see that each of these graphs represent a subject, and for example, in this analysis, they were testing for the performance with respect to some tasks, like mathematical task, for example. And as you can see, this topological summary is able to capture a very important aspect. So the right-hand side exhibit some modularity with respect to the task. So, for example, working memory or math or video, they stimulate different activation patterns, while the low-performing subject seems to have the same area of the brain, which is doing different tasks. So being less specialized, indeed, it performs worse. And it's amazing that we are able to understand this with a glance. So what is the pipeline of DyNeuSR-based analysis? First thing, of course, we load our data, one subject, and we will not delve into preprocessing. Second step, it's optional, but actually it's very important for performance and to avoid the curse of dimensionality. We can reduce the dimension of a single time frame from many pixels to a few numbers in basically two ways based on neuroscience. So we can do some masking. So we just extract some voxel from an area of interest, or we can use parcellation by averaging neural activity in brain areas. So using an atlas of the brain, or we can use some methods drawing from data analysis of BCA, ICA, and so on and so forth. So once we have our reduced data, we can apply mapper to the following point cloud. One point is associated to a time frame of our scan. And then we can generate these DyNeuSR visuals using, for example, different colorings or labels that enrich our comprehension of the graph. So let me give a very brief review of mapper. We start with a point cloud, and we do 4 phases. So, for example, if we start with the sampling of a trefoil in the 3D space, and this is the classical tricoloration of the trefoil. First step is filtering. So we project the points to a very low dimensional space, like 1-dimensional or 2-dimensional. This is different from the dimensional reduction that we do Before that, maybe it's like a 50 dimension or 100 dimension. It's just a pre-filtering, let's say, but this is intrinsic to the algorithm. Second thing is binning. So we now divide these projective points using overlapping cubes. And third step is clustering. So we take the pre-image of each of these cubes, and we cluster the points in the original space. In each bin separately. And once we have these clusters, we can finally draw our shape graph. So vertices are clusters that we found in each bin, and two clusters are connected by an edge if they intersect. Recall that bins are overlapping, so some points can be shared between a cluster of a cube and the cluster of the cube right next to it. And as I mentioned, in our neuroscientific context, each point will correspond to a time frame. Okay, so let's see in practice how this works. Unfortunately, the current version only works with Python 3.6, so I feel free to pause the video and copy these commands to get working Conda environment if you use Conda. If you use pip, please feel free to visit this GitHub repository that contains all the necessary instructions and all the examples of code that I'm showing in this presentation. And if your editor does not handle Python 3.6, for example, the latest version of Spider doesn't, I suggest you to use PyCharm. Okay, so let's see a real example. This is the Haxby dataset, and as I mentioned, there are four phases that I've highlighted. So the first one is just Haxby is actually available online, so you can fetch it directly from Python. And then we apply a mask. This is the dimensionality reduction. What does it mean? We just extract voxels that come from ventral temporal cortex. And there is a reason why we are doing this that I will show you in a minute. And this is actually more about the fourth phase. So we encode labels as integers. What does this mean? We want our graph to show us which task the subject was performing at each given time frame. This is the third phase, so generate the shape graph using Kepler mapper. Yes, you can see there are many actually parameters that one can choose here, and this is what really takes some time to master. For example, which projection use for the first phase, for the filtering case, how many cubes we use. This 20 is how many cubes we use for each axis. 0.5 is the percentage of overlap between cubes, and this is the cluster, which can be actually very different. This is dbScan, and then we have to choose a parameter for say the cluster resolution. So it can take some time to choose the right parameters, and you will develop some intuition, but on the other hand, it also gives a lot of flexibility. And the last phase, we can actually generate very easily our DyNeuSR. For the packages to import, please visit the GitHub repository for a working code. And this is what you get. This is an object recognition task. So as you can see here in the legend, we have chair, barrel, scrambled pigs, house, show, cat, face, scissors, and rest. And this is our dinosaur representation. And it is known that the ventral temporal cortex encode different kind of visual stimuli in different ways. And it's very interesting that this topological summary is able to capture this. So look at this. The resting state patterns are very, very close to each other. And then we know that landscapes like house are in a different encoding side. And here we have green is face and red is cat. And indeed, face recognition is another sort of stimulus. And we can see, for example, in the middle, we have tools and objects, and the scrambled pics, which is just random pixels. Its noise is another group of vertices. So we are able to see at a glance a very important and deep result in the study of ventral temporal cortex. So to wrap it up, just let me notice that if you don't do this extraction, but for example, here I did a PCA, as you can see, the result is very different, very different. But it's still interesting because like I can see that rest, the blue, is on many, many vertices. So for example, from this summary, I could conjecture that the resting pattern could be sort of daydreaming, and I want maybe to project orthogonally to this activation map, and I could get some results more specific to the image recognition. So even if we hadn't the neuroscientific knowledge about the ventral temporal cortex, we could still come up with some conjectures and then perform some more quantitative analysis. So thank you so much for your attention. I hope you enjoyed and good luck with your wonderful research.